Hi, I'm Danny Winder. I'm at Vanderbilt University at the Center for Addiction Research here and in the Department of Molecular Physiology and Biophysics. And I'd like to thank the organizers for inviting me to participate in this conference, and look forward to talking on the panel later. But first, look forward to the opportunity to tell you about some of the recent work in my lab and collaborations with another lab that we've been working hard on. So that with that, I'll pull up my slides here and we can dive in. So one of the major foci of my lab is in understanding mechanisms underlying alcohol use disorders. And we know that alcohol use disorders are a major public health problem in our country, with over 95,000 deaths annually from alcohol-related causes in the United States, and nearing 50 million people a year who could be diagnosed as having an alcohol use disorder at any given time. So this is a significant health issue, and our lab is interested in understanding mechanisms that underlie these processes that produce this disorder, so that we might take advantage of that knowledge to develop new therapeutic opportunities. So we, like many in the animal model field, seek to develop animal models towards aspects of disorders like alcohol use disorder. And what I want to show you today is some of our efforts in my lab to do that, and then our efforts to work together with human imagers, human brain imagers, to test our hypotheses that we generate from our animal models in a construct validation approach in human imaging, in the human imaging realm. And then our efforts to seek to further refine our models based on that input and to extend that knowledge. So this is a typical conceptualization of alcohol use disorder in the field, generated by George Koob and colleagues. And it centers on the idea that alcohol use disorder has multiple potential entry points, and is a cycle here that is used to conceptualize alcohol use disorder, beginning with a preoccupation anticipation stage, followed by binge intoxication, and then moving through to a negative affect and withdrawal phase. And this is the phase that my lab is particularly interested in. We think that this is an important therapeutic barrier because this is the phase that emerges during treatment and abstinence. And so the depression, anxiety, disrupted HPA axis, all of these features that emerge during prolonged withdrawal and abstinence are stimuli that may drive negative reinforcement-based relapse behavior. And so we're interested in understanding mechanisms that drive this part of the AUD cycle. So we've been utilizing an animal model in my lab in the last few years. That's a very simple one. We singly housed C57 Black 6J female mice, and then we allowed them long-term access in their home cages to alcohol or water. So they have two bottles in their cage all the time, one a water bottle and one an ethanol bottle. And this occurs for 6 weeks. And after 6 weeks, we remove the alcohol bottle and replace it with a second water bottle. And that then moves into a forced abstinence period. So chronic drinking, forced abstinence, or CDFA. And so you can see during the chronic drinking phase, these female mice develop a high preference for ethanol and the ethanol bottle compared to the water bottle, and will consume substantial quantities of alcohol, particularly from weeks 3 on in this chronic drinking phase. It's the forced abstinence phase that we're particularly interested in, and in the idea that negative affective phenotypes might emerge during this forced abstinence phase. We unfortunately obviously can't ask the animals how they feel, so we use a series of simple behavioral tasks to assess the animal's behavior and to make assumptions on phenotypes that emerge. One of these is a very simple task, the forced swim test. This is a pharmacologically validated test where the immobility time in this forced swim test is regulated by antidepressants. So clinically relevant antidepressants will decrease the immobility time of animals in this task. And what we find in this forced abstinence phase is that, interestingly, in the first day of forced abstinence, there's no phenotype in the forced swim task. But if we look out a couple of weeks into abstinence, we find that a phenotype emerges. So in the ethanol animals compared to sham animals, there's a big increase in the immobility time of these ethanol abstinent individuals. So this is interesting to us because it suggests that behaviors emerge during the prolonged abstinence of the availability of ethanol. Another simple task we use to converge with tasks like the forced swim task is the novelty suppressed feeding task, or NSFT. In this task, the mouse is briefly food deprived and then allowed access to a brightly lit novel space in which is placed in the center a food pellet. And then what we measure is the latency for the animal to begin to eat that food. And that latency is a pharmacologically validated value. Anxiolytic compounds or antidepressant compounds will decrease that latency. Stressors will increase that latency. And as you can see here, again, comparing control animals to forced abstinence CDFA ethanol animals, we see there's a large increase in this latency that is out here in the 2 weeks of forced abstinence. And this is quite persistent in nature. So in this cohort of animals, we waited a month after the chronic drinking was completed and still saw a substantial change in the latency. So these data suggest to us that animals that undergo chronic drinking forced abstinence enter into a negative affective-like state that may be relevant to anxiety and depression. And so just to summarize, with the CDFA model, female C57 black 6 mice consume large quantities of alcohol when they're isolated in their home cages. And forced abstinence from this alcohol access results in behavioral disturbances that may be related to anxiety and depression, and therefore may be related to this negative affective withdrawal phase that we are interested in. So to begin to explore mechanisms that underlie these alterations in behaviors, we became interested in exploring this novelty suppressed feeding task more, as well as the forced swim task, and understanding what neural circuits contribute to these differences that emerge in these behaviors. You can see in the brain of this cube image of alcohol use disorder that these particular brain regions are heavily implicated in the negative affective component. And so the bed nucleus of the stria terminalis, or BNST, and the central nucleus of the amygdala, or CEA. These two structures from a large literature have been implicated in this negative affect withdrawal phase of alcohol use and substance use disorders. The BNST is a region of the brain that my lab has been particularly interested in for many years now. In the human brain, it spans the anterior commissure. It's a deep basal forebrain structure, and the BNST is about the size of a sunflower seed in the human. In the rodent, it has a very similar localization. It again is a basal forebrain structure and surrounds the anterior commissure here. This gives you a sense of one of the reasons that the BNST and central nucleus are part of what's referred to as an extended amygdala. This is work from Mike Davis and Walker from many years ago, in which tracers were placed in basolateral amygdala, and then output regions were looked at. So this is an anterograde tracer that was placed into the posterior portion of the basolateral amygdala. And you can see, as you would predict, that there are large projections to the central nucleus of the amygdala, but then you can also see here equally large projections to the BNST. And so the BNST and the central nucleus together are two of the very largest outputs of the basolateral nucleus. And so my lab's been studying the BNST and the actions of acute and chronic alcohol on BNST function in rodents for over a decade now. And this model summarizes the conclusions of many of those studies. We're interested in the idea that glutamate inputs to BNST neurons play an important role in driving relevant BNST-dependent behaviors. And our evidence points to the idea that these glutamate synapses are upregulated during alcohol withdrawal and alcohol abstinence, that there's an increase in excitatory drive from other brain regions onto BNST neurons, thus activating them to initiate circuit-dependent behavioral adaptations. And one of the questions that we've been very interested in is trying to understand which glutamate synapses, what are the external inputs to the BNST, whose signals are upregulated during alcohol abstinence. And this is another way of looking at those inputs. I showed you the older data from Davis's group with an anterograde tracer. This is a modern look at the inputs to the BNST using viral-mediated tracing approaches. Here we simply use a retrograde adenovirus. We infect BNST neurons specifically with this adenovirus, wait a period of time during which that virus is taken up by regions of the brain that project to the BNST. And then we process the brain using a brain clearing approach so that we can image the entire brain using light sheet microscopy. And so this gives you an idea then of the various inputs to this BNST region. So you can see the BNST highlighted there. You can also see this large stria terminalis now in 3D. This is the white matter bundle that connects the BNST region shown here with the amygdaloid complex. And so as we spin around here, you'll see the stria terminalis spanning up and then back down into the basolateral amygdala, as well as the central nucleus of the amygdala. And so this interconnectivity features importantly in a variety of things, such as generalized anxiety, fear conditioning, et cetera. We became very interested in some of the cortical inputs onto BNST neurons, and in particular, this population of cells here within the insular cortex. The insular cortex is a hidden cortex in the human brain. Here it is in the rodent brain, and it has been implicated in a variety of behaviors relevant to alcohol use disorder. The insular cortex is an integral brain hub that connects different functional systems to regulate interoceptive states. It plays roles in reinforcement learning and decision-making and emotional control. And so we became interested in the idea that the insular cortex projections into the BNST may play a role in some of the alcohol use disorder related phenomena that we're studying, and we wanted to explore that. If that synapse is involved in the behaviors that we've been studying, then one would predict that if we specifically activate that pathway, we should be able to recapitulate behaviors like this novelty suppressed feeding task behavior that we observed during forced abstinence. To test that idea, what we did was to use a viral strategy to selectively deliver a switch to the BNST cells that receive insular input. And so the way we did this was to use two viruses, one that we injected into the insula and one that we injected into the BNST. In the insula, we put a special AAV1, which is an anterograde Cree. So this is a Cree that it expresses, it infects the cells in the insula, moves down the axon, and it's capable of anterogradely jumping synapses and moving to downstream neurons in a neural circuit. So this then delivers a molecule called Cree recombinase, which then enables the expression of a switch that we delivered by another virus into the BNST. So the BNST virus, for its product's expression to occur, needs this Cree to be present. And this switch is a designer receptor exclusively activated by a designer drug, a DREAD. This is a chemogenetic strategy in which we're expressing a protein specifically in these cells that can control the activity of those cells when a specific drug is provided. And that drug is clozapine N oxide, or CNO. So delivery of CNO to the animal activates this specific flavor of DREAD, the HM3DQ, which it does many things inside cells, but one thing that it does is it tends to increase the activity of those cells. And so consistent with that, we measured an immediate early gene, CFOS, and found that when we give the animals that have been infected in this way, clozapine N oxide, we saw a big increase in FOS expression in infected cells in the BNST, consistent with their activation. Importantly then, in those same animals, we saw a big increase in that novelty suppressed feeding task latency. suggesting that in this sufficiency experiment, that activation of BNST neurons that receive input from the insula can produce an NSFT phenotype similar to what we observe in the abstinent state in CDFA. So the insula to BNST input is sufficient to produce the behavior. Is it necessary to produce the behavior during forced abstinence? One hypothesis, if that were the case, would be that we would see increased activity of the insula to BNST pathway. And we tested this in a couple of different ways. First, we looked at the BNST in forced abstinence in a couple of different approaches. First, we looked at that FOS molecule again, a crude marker of neuronal activity. And you can see that in this forced abstinence phase, there's more FOS expression in the BNST compared to sham water control animals. And if we patch onto individual BNST neurons in a brain slice and then record excitatory drive from synapses like those insula synapses onto these cells, we see that there's increased excitatory drive onto these cells as well. So FOS-based increase in activity and appears to be an increase in glutamate input onto these cells. But what's driving this increased input in each of these cases? To test the role of the insula in these processes, we repeated the dread-based strategy, but this time expressing the dread in the insula and expressing a different flavor of dread. So now we're expressing HM4DI instead of the HM3. And the DI version, the GI-coupled GPCR, tends to inhibit transmitter release. So we express the HM4DI in insular neurons. It goes down the presynaptic terminals into regions such as the BNST, so that when we activate the GI GPCR with CNO, we can see its impact on BNST function. And so we do this in brain slices where we're again recording from individual neurons in the BNST. Sam Santani, the postdoc in the lab at the time who did this work, again saw an increase in excitatory transmission in our ethanol-treated animals compared to our water-treated animals. And he saw that if he added CNO in the solution that bathed the brain slice, that he could reduce this spontaneous transmission in the abstinent animals, but not in the water control animals. And so this tells us that some of this upregulated excitatory transmission that we see in the BNST indeed involves insular synapses as a source. And corollary at the whole animal level, we repeated the FOS experiment, now activating that insular GI dread to decrease that terminal activity in the BNST. And in concert with what we observed with excitatory transmission in the BNST, we see that activation of the insular inhibitory dread decreased the expression of FOS in the BNST, suggesting that that increased FOS expression during abstinence is in part driven by upregulated insular input on these cells. We then moved to the behavioral level to assess whether inhibition of this circuit utilizing the GI dread would normalize the abstinence-induced phenotypes that we observe in the forced swim test and in the novelty-suppressed feeding task. And so on the left are a variety of control experiments. So I'll call your attention to the data on the right, which is the data for the NSFT task and the data for the forced swim task during abstinence. And you can see that in both cases, animals that were given CNO, we saw a reduction in the elevated negative affective-like behaviors that we saw in the forced swim and in the NSFT. So in summary, insular inputs in the BNST in mice is upregulated during alcohol abstinence, and Insula-BNST interactions participate in behavioral adaptations during CDFA. So we are excited by these data. We think that our animal model has yielded potentially useful synaptic information about parts of the brain that are interconnected that may regulate key components of behaviors that emerge during alcohol abstinence. But we wanted to know whether we could construct validate this phenotype that we observed through collaboration with human imaging research. And so we're really fortunate to have as a collaborator Jennifer Blackford. Dr. Blackford, during the initial period of this collaboration, was a professor in the Department of Psychiatry at Vanderbilt University here and a member of our Center for Addiction Research. She's now the director of research at the Monroe Meyer Center at the University of Nebraska. And she is an international expert in the human BNST. So I mentioned that sunflower seed-sized nucleus in the human brain. Dr. Blackford's lab developed initial pulse sequences and masks using a 7 Tesla MRI scanner to reliably detect the BNST and associated brain regions. They then developed pulse sequences where they could utilize a 3 Tesla magnet to enable them to scan a larger number of subjects and monitor the BNST and connections with other brain regions. These masks and pulse sequences that they developed have been widely shared and validated amongst a variety of labs in the human imaging field. So it's been a privilege to get to work with Dr. Blackford on this and to continue to work with her. So we were fortunate to receive funding for this work through an R21 from NIAAA. And Dr. Blackford recruited a very talented MD Ph.D. student, Lizzie Fluke. Lizzie had trained with Christophe Grion and Monique Ernst at the Intramural campus and already had experience in human BNST and anxiety-based research. And so we were excited to bring her on board. She spent quite a bit of time establishing baseline measures of the human BNST and the human insula in particular, verifying that she could observe structural connectivity between the human insula and BNST before moving on to an alcohol use disorder cohort. Lizzie and Jenny then recruited an alcohol use disorder cohort in early abstinence in this initial R21 funded pilot study. We ended up with 20 healthy controls and 19 abstinent AUD subjects. You can see the audit scores, which are used to establish relative alcohol use disorder, much higher in the abstinence group compared to healthy controls. And then, as we'll talk about in a bit, there were clinically significant anxiety and depression scores amongst this abstinence group as well. Lizzie and Jenny then began by examining structural connectivity between the BNST and the insula in this abstinent state compared to controls. And as you can see here, the original data, when collapsed, did not reveal highly significant results. You can see that the control groups and the abstinent groups bending across an anterior insula and a posterior insula didn't show much in the way of differences. However, when they separated the data between men and women, something very exciting to us emerged, which was that in women, a correlation existed between structural connectivity or greater structural connectivity in the abstinent population compared to the control population, both for the posterior insula to BNST and the anterior insula to BNST, something that was not really seen in the males in this cohort. Moreover, as I mentioned earlier, there was quite a bit of difference in anxiety symptoms and scoring in the abstinent group relative to controls. This is not surprising given the alcohol use disorder conceptualization that I showed you at the beginning of the talk. And this was also similar for depression scores, quite a bit of difference in the AUD cohort compared to controls. And so we were really interested to learn that there was a strong correlation between anterior and posterior insula connectivity with the BNST and these depression scores in particular that were observed in this cohort. So in summary then, in the human data, Insula-BNST structural connectivity correlated with depression scores in alcohol abstinence. And data indicate increased structural connectivity between the insula and the BNST in abstinence in females in particular. And this is of great interest to us because you'll recall that in our animal model, it's in fact an animal model that is restricted to females. And that's because in our home cage model, females drink much, much more alcohol in this inbred strain than the males do. And so the forced abstinence phase becomes confounded because there's so much less alcohol on board in the males of this inbred strain compared to females. So given this hint from the human work that we may be on the right path here with studying the Insula-BNST projection in the context of the development of negative affect during alcohol use disorder development, we wanted to start to think about approaches that we might take to think about translation with. And so we began by utilizing commonly explored novel antidepressant ketamine, which is now widely explored both at the animal model level and in humans for its novel actions as an antidepressant. And we examined the impact of acute ketamine administration on both the forced swim test behavior that emerges during forced abstinence and on the novelty suppressed feeding task behavior. And here you can see that if ketamine is administered right before we do the novelty suppressed feeding task, we see that the ketamine dramatically reduces that latency increase that the ethanol abstinence produces, suggesting that this antidepressant has intriguing efficacy in reducing the symptoms that emerge during abstinence. In thinking about this, though, we wondered if there were more translationally clever approaches we might take than trying to treat symptoms that have already emerged. So as I mentioned early on in the discussion of the animal model, an interesting feature of the forced abstinence phase is that the anxiety depression begins at a normal state at the initiation of forced abstinence and then grows over time during this 2-week window. So seeing very little in the way of depression-like phenotypes in this early phase, and then the emergence of these phenotypes a couple of weeks later. So are there ways that we could intervene here in interesting ways to produce more interesting outcomes from a potentially clinically relevant perspective? And one way to think about this is to think about it from the internet meme approach of how it started, how it's going. And to give an example of what we're thinking about, this would be a typical way this meme is utilized. And this is actually the anniversary or near the anniversary, the day that I'm recording this, of tornadoes that hit Nashville in 2020, right before the beginning of the pandemic. And this is the aftermath of that tornado in one of the iconic buildings in the East Nashville region of the city. And so this would be the how it's going. It was the idea that this building was demolished by a tornado. And this is how it started. It was a tornado that came through that destroyed the building. So thankfully, many of these buildings have been restored in recent years, in the last 2 years. This is an example of that same building the night of the storm, and then a year later, completely restored and open for business. And I would equate this to the ketamine data that I just showed you. So we can reduce symptoms with acute administration of ketamine. We can restore this building, but this does not reduce the likelihood that another tornado could come through and then demolish the building. So what if we thought about, instead of treating the symptoms by rebuilding the building, what if we could somehow control the likelihood that the tornado is going to come through or weaken the impact of the tornado to begin with? And so with this in mind, we designed a different approach to this study where instead of giving the ketamine immediately before the behavioral task, we gave the ketamine at different windows during forced abstinence and then examined the behavior that emerges during forced abstinence. And we observed an interesting outcome, which was that if we gave ketamine right at the onset of abstinence, we prevented the development of the depression-like phenotype that emerged 2 weeks later. So in each of these bar graphs down here, we've given ketamine at a different time point, but in each case measured for swim at the same time point 2 weeks after the initiation of forced swim. So each of these is a separate cohort of animals. And so these data suggest a very interesting possibility that there's a critical period at the initiation of forced abstinence where you could inoculate the animal or individual with a pharmacological intervention that might prevent the manifestation of some of the behavioral consequences that emerge that may drive subsequent relapse behavior. And so this was a really exciting outcome to us and suggested that we need to really focus in on this initial window, this emergence period at the beginning of forced abstinence, and try to learn more about what's going on during this early period. To do that, though, it still requires going through 6 weeks of chronic drinking to get to this very short window of time. And we have a number of mechanistic questions that we'd like to ask about how these circuits are regulated. So we started thinking about more rapid screening approaches we might take to learn about the acute effects of stress on some of these key synapses so that we could then test key questions with the full chronic drinking forced abstinence model. And we, based off the knowledge that the acute withdrawal phase is in itself a stressor, we started thinking about looking at some of these same circuits in the context of a stressor. And we chose to utilize a very well-studied and well-described form of stress, which is restraint stress. Here you simply use a conical tube and place a mouse in that tube with the tip cut off so it has ready access to air. And so animal is then immobilized for a brief period of time. This produces reliable recruitment of the HPA axis and behaviors consistent with engagement of stress responses in the animal. So our first question was whether restraint stress produces engagement of the circuit that we're interested in studying. Does it induce activation of the Insula-BNST circuit? And so to ask this question, Tracy Federley, when she was a grad student in the lab, performed an experiment in which she placed that inhibitory dread, the GI dread, in the insula on one side of the brain, and then placed a control for just GFP, on the other side of the brain. She then let the animals recover for 3 weeks, gave them the dread drug, CNO, to activate that GI dread and inhibit the insular input to the BNST on one side. She then restraint stressed the animal for 60 minutes. 30 minutes later, processed the animals for FOS immunoreactivity. And what she observed was that certainly, as we would expect, if we measure FOS again, that immediate early gene, that's a crude measure of activity, FOS expression was increased in the BNST in stressed animals relative to control animals. In the dread expressing animals, when a CNO was administered, she saw a reduction in BNST FOS expression on the side of the brain where the GI dread was expressed, consistent with the idea that the BNST is engaged by restraint stress, and at least some of that engagement is coming through insula-dependent activity and activation of BNST neurons. So what is happening during restraint stress? The animals really have two choices that they can engage in restraint stress. They can either struggle, they can engage in physical bouts of activity, likely aimed at escaping, or they can be passive and remain still. And so in the early 2000s, this was described as an active and passive coping approach that the animals could take. And Satya Patel and C.C. Hiller demonstrated that blocking CB1 receptors, which would increase anxiety state in the animals, increases the active coping bouts that these animals engage in, more physical struggles while in restraint stress. And then Nicola Grissom also showed that if an animal was stressed prior to being placed in the restraint tube, this also increased these active coping bouts, if you will, or these escape attempts that the animal engaged in. So these active activity events that occurred during the stress are something that we are interested in studying and looking at how activity in the insula and BNST correlates with those active coping events. And so to do this, Kelly Williford in the lab developed a novel device that we could utilize to measure activity in the brain while an animal engages in restraint stress. This is a restraint tube, as she called it, recording signal transients accessible in a tube. So the key adaptation being a slot cut into the top of this device so that we can get a fiber into a chronic implant on the animal's head so that we can measure light coming out of the brain and push light into the brain for photometry, calcium imaging, and sensor studies. And so Joe Lusinger and Sam Santani embarked on a large project where they measured the activity of neurons in the BNST and in the insula while animals engage in these active coping events during restraint stress. And so this involved the combination of a machine learning-based tracking of activity using deep lab cut, so software that can be trained to identify activity events when they occur and to timestamp those events. And while this is going on, using fiber photometry to measure the light emitted from fluorescent sensors that are excited by light pushed through our fiber onto these sensors that we've placed in the brain through viral expression. So we can measure the light coming from these sensors as the animals are engaging in these active coping events. And these are just some examples of some of the data that we generated, first looking in the BNST, and here looking specifically at CRF neurons, a special unique population of neurons in the BNST. On the top here, we're looking at repeated active coping bouts stacked one on top of another. So this is the number of bouts going down here. And then at the bottom is the bulk average of all of these events compiled together. So you can see that at time 0 is the beginning of one of these active coping events. And you can see that if we just express GFP, a fluorophore whose fluorescence is not dependent on any sort of activity, we don't see a large response here. And so this is an important control for physical changes that could occur in the light path or something like that, artifacts of movement. However, if we express the molecule GCAMP, which is a fluorescent reporter of calcium in an individual cell, we saw that the active coping events were associated with large calcium transients. Further, consistent with the involvement of a glutamate synapse and that insular input to the BNST, if we used a fluorescent sensor that senses the presence of glutamate, and this particular sensor is called a glue sniffer, we see that there is also a large glutamate transient associated with these coping bouts. The hypothesis being thus that glutamate released from inputs such as the insular input to the BNST is driving this calcium signal in BNST neurons. And so as one way to test that, we moved upstream and performed similar experiments now where we measured the activity of neurons in the insular cortex and neurons that project to the bed nucleus of the stria terminalis. And we used a variety of sensors again. We used the calcium sensor GCAMP to measure relative activation of neurons in the insula. We used glue sniffer to look at the presence of extracellular glutamate during these events. And then we also used a GABA sniffer to measure the availability of extracellular GABA during this time. Again, time 0 being the initiation of one of those active coping events as defined by Deep Lab Cut, the machine learning-based software. So you can see that there is a substantial calcium transient in these insular cells that occurs with the onset of these active coping bouts. This is mirrored by a glutamate transient in these cells. And then very interestingly to us, we saw the converse of that, that GABA was transiently decreased at these synapses. And I'd be happy to talk about that more during the panel discussion, perhaps. So this suggests that this insula to BNST circuit is highly responsive to physical activity and motor events in the animal. And this was a surprise to us because it wasn't clear to us how these Insular-BNST circuits would get motor information. Where is this input coming from? To try to determine or answer this question, we began by using a viral tracing approach. So we used an approach called TRIO, which is tracing the relationships of inputs and outputs. Basically, this is a way to determine all of the inputs to a very specific population of neurons in the brain. And so what we did was to express some viral constructs in the BNST and some viral constructs in the insula to recapitulate a rabies-based tracing system, which would then be retrogradely delivered to the neurons we want to find, those neurons that project to insular neurons that go into the BNST. Okay? We can then use our light sheet imaging approach again to examine those cells. And then we can use, again, a different type of machine learning-based software to actually quantify these individual dots as neurons and register them to a digital mouse brain atlas so that we can get a readout of the brain regions that project onto these very specific cell types. And in the interest of time, I won't play all the way through this movie, but you can see that there were cells throughout the brain, unsurprisingly, that project onto this population of neurons. So then these are some of the very top hits of regions that project onto those insular cells that go to the BNST. And I'll call your attention in particular to the somatomotor areas. This was a big surprise to us that the insular cortex gets a large input from motor and premotor areas, areas involved in the executive control of movement. This is probably to no surprise to people in the motor cortex field, because there's a concept known as efference copy, first defined in 1950, that individuals working in the motor cortex field have known about for a long time. And so motor commands that originate in the motor cortex obviously go out and initiate actual motor actions through engagement of appropriate muscle sequences. But these motor commands that go out to initiate those physical activity events also are involved efference collaterals that go to widely divergent parts of the brain to convey that this information, that these motor events are happening. So the event is sequenced by premotor and motor portions of the brain, and then it's also broadcasting to the rest of the brain that this is happening. And so there are a variety of examples of where this efference copy mechanism has been shown to be of importance. One would be the idea that you can have a smooth visual field while you're walking or moving in some way. And that's because your motor cortex is telling your visual cortex about planned movement, so the visual cortex can adjust the visual field accordingly. Similarly, a person can't tickle themselves typically because of motor cortex direct innervation of somatosensory cortex that tells somatosensory cortex that movements are incoming. And so there are lots of examples of this. And what we're excited to explore now is what are the roles of efference copy for motor cortex inputs to regions of the brain like the insula and the BNST that are involved in interoceptive processing and negative affective-based behaviors. And so we've become interested in the idea that exercise through engagement of this efference copy mechanism might be an attractive strategy to explore in manipulation of negative affective disturbances and manipulation of alcohol intake. And this is recently published work from our lab where we utilize exercise wheels in the chronic drinking forced abstinence assay. And on the left here, you can see a correlation of the wheel rotations per day relative to the relative ethanol preference that these animals have. And you can see that there's a very strong correlation in overall ethanol preference relative to wheel rotation, where the more time the animals spend on the exercise wheel, in general, the lower the preference that the animals have for alcohol, which was a very interesting result to us. And then here are data in which animals that went through CDFA were given exercise wheels in one of two ways. One, an unlocked wheel to serve as a control for, one, a locked wheel to serve as a control for the novelty of just having the exercise wheel device in the cage, and the other, an unlocked wheel where the animal can actually engage in exercise. And you can see that in the unlocked wheel state, the latency for the NSFT task that increases in abstinence was substantially reduced in the animals that had access to these exercise wheels. And so we're very interested in continuing to pursue this potentially interesting link. So in summary, struggle events during restraint stress are monitored by an Insula-BNST circuit. Glutamate reporter analysis suggests that this monitoring involves extended amygdala circuitry. Motor and premotor cortical regions provide substantial innervation of BNST projecting insular neurons. And these data provide evidence for a feed-forward mechanism by which motor planning may inform interoceptive and affective circuitries. And so we think that these trio data in particular not only allow us to not look under the lamppost and find these interesting possibilities to explore, like the motor efference copy model, but they essentially provide us with a control network for an alcohol abstinence affect circuit. And so this is a control network that we've delineated in the mouse model. We now look forward to working with Jenny to push this back into the human and to ask about the relative likelihood that there are alterations in any of these control networks with particular endophenotypes associated with alcohol abstinence. And so we're excited to continue those studies with Jenny, and she has a newly funded R01 from NIAAA to begin to do so. And so with that, I'll stop and thank our very generous funding sources, a great lab, many of whom I've mentioned at various times throughout the talk today, and a series of collaborators who've helped us with these and other studies, and in particular, Jennifer Blackford's lab with the human imaging work, and Lily Fluke, an MD Ph.D. student in her lab. So thanks very much for your attention, and look forward to chatting during the panel discussion.